We're on chapter number 24 now. In this chapter, we're going to look at the chromosomal theory of inheritance. So recall back to the previous chapter, we talked a lot about Gregor Mendel with his Ps. So in 1865, Mendel called genetic material elementen. This was in his seminal paper that, of course, predated the understanding of chromosomes and genes, but he basically got everything right in terms of understanding that there were these essentially packets of information, which he called elementen. So a little bit later, around 1902, Sutton and Bovary and others independently observed chromosomes of a cell under a microscope. According to them, Mendelian genes have specific loci along chromosomes. Loci is simply plural for locus. And it is chromosomes that undergo segregation and independent assortment. This is the chromosome theory of inheritance. So let me draw a little picture here. So you can see here in the photograph, we have some DNA that's been stained, and they're showing you in yellow. There's some sort of tag showing you the locus of a gene. So let me just as a reminder, let me see if I can draw a picture here. Nope. I need to grab. There we go. All right. So, okay. So imagine this is a gene. We have a pair of homologous chromosomes. Remember that you have 23, as a human, you have 23 homologous chromosomes. And on a chromosome, there are genes, and the genes are located at various loci. So what I've drawn here is, let me go back to the original color. The colors are strange here. So one location is called a locus. Okay, and if you have multiple genes, you refer to these as various loci. So the idea is that at this locus, you would have a specific gene, for example, one for eye color, or if we were talking about Ps, whether the P itself was either green or yellow, whether the pod was wrinkly or smooth, that sort of thing. The next idea we're going to look into is gene linkage. This is the idea that a single chromosome contains many alleles in a definite fixed order. And this fixed order occurs because each allele has its own location on the chromosome called a locus, which I mentioned earlier. So what is this idea of gene linkage? Well, the idea is that all the genes on one chromosome are essentially linked together because they're on a single chromosome, and they're going to tend to be inherited together. So I realize that this goes against what I talked about with Gregor Mendel, but with Mendel, it was very likely that the genes that he happened to pick, he was very lucky they happened to probably be on separate chromosomes. Now, if you have two genes that are on the same chromosome, then you don't have independent, they won't be segregated, independent, and you won't have independent assortment. They'll tend to go together. So this is going to affect things like, for example, well, again, so his two trait crosses, the ones that we did earlier, he's assumed that genes are on non-homologous chromosomes. Genes that are linked do not show independent assortment. Now, we're going to get into a little more detail on that, but in general, if the genes are on the same chromosome, and specifically if they're close together, and we're going to get into that in just a second, then they may be linked pretty tightly, meaning that they'll get inherited together. In this slide, we see a number of genes that are linked together because they happen to all be on the same chromosome. So the human chromosome number 19. Remember that you have 23 pairs of homologous chromosomes, and so they're numbered. And actually, if we look at the karyotype, recall that the number 1 chromosome was the longest, and so they get progressively shorter. And then that's going through all the autosomes. You have 22 pairs of autosomes, and then we have a final pair of sex chromosomes. So this is an autosome. And here we have a bunch of diseases. So recall the gene itself is not, we don't have genes obviously for diseases, but these locations are locations for genes. And if there's something wrong with a gene, then we have these disease states. So the first one here is muscular dystrophy. This is a progressive weakness and loss of muscle mass. Next one is, and there, you can see these are all in order. Okay, so let me see if I can point at these. So yeah, I'll use the arrow here. Yeah, so the first one we read about here. Then we have atherosclerosis. Susceptibility. This is a buildup of fats and other substances in the artery walls. It leads to atherosclerosis. Literally means hardening of the arteries. Next up, we have insulin-resistant diabetes mellitus. In this case, we have body cells that don't respond normally to insulin. So insulin is a hormone that's released by the pancreas, and you need it because it helps cells in your body to take in glucose. So if you have a meal, the glucose level in your blood spikes, and that's actually kind of dangerous, and you want to bring that glucose into your the cells of your body. Unfortunately, if the body cells don't respond normally to insulin, these cells throughout your body won't pick up the glucose. So the glucose will stay relatively high for a long period of time. And actually, diabetes mellitus, let me see if I can use the highlighter here. So this mellitus here, this actually refers to the idea that the urine is actually sweet because it has excess glucose in it. Okay, the next one is ichthyosis. This is a group of skin disorders characterized by dry, scaly, or thickened skin. Next up on the same chromosome 19, we have eye color, whether you have, for example, green or blue eyes. Next is hair color, for example, brown hair color. The gene for that is there. Acute myeloid leukemia is a type of cancer of the blood and bone marrow with excess immature white blood cells. So unfortunately, what happens is you have these excess immature white blood cells, and because your bone marrow, of course, makes red blood cells as well as white blood cells, these sort of crowd out the white blood cells that you do need, as well as the red blood cells that you need to carry oxygen. So people with acute myeloid leukemia would be very weak, and they would also be susceptible to getting sick. Alzheimer's disease, late onset. This is the more typical type that often people start to get as they age, perhaps beyond, say, age 65 or so. It's a progressive disease that destroys memory and other important mental functions. The next, we have a gene for deafness. So essentially, if there's something wrong with this gene, then people end up deaf. Next, we have hemolytic anemia. So let's look at this word hemolytic. So hemo refers to blood, and lytic, you've heard the term lysis before. So lysis is to break apart. So hemolysis is the breaking apart of red blood cells. And anemia is when you essentially, there's different types of anemia, but anemia is a form of disease in which you're essentially not getting enough oxygen to your tissues. And so people tend to be very tired and weak all the time when they have anemia. So in hemolytic anemia, what happens is the red blood cells are destroyed faster than they can be replaced. Next one is ovarian carcinoma. A carcinoma is a cancer that forms in epithelial tissue. Colorectal cancer, cells in the colon or rectum grow out of control. So the colorectal cancer is happening at a higher rate now in younger people. It used to be years ago that you would get your first colonoscopy around age 50, and now the recommendation is you should get your first colonoscopy around age 45. Cardiomyopathy. Cardio has to do with the heart. Myo refers to the muscle, and then this pathy means pathos, which refers to a disease state. So it literally just means a disease of the muscle of the heart. So this is really a collection of diseases of heart muscle. Okay, so earlier I mentioned the idea that if genes are on the same chromosome and they're located close together, then they tend to be linked. They tend to be inherited together. And in fact, all the genes on a single chromosome are said to be in a linkage group. However, if the genes happen to be fairly far apart on an individual chromosome, it is possible that you can have them separate when we have the formation of the gametes. How does that occur? Well, During meiosis, there's a phenomenon called crossing over that sometimes occurs between non-sister chromatids and a tetrad, and this leads to exchange of genetic material. We actually talked about this earlier when we covered this meiosis back, I believe, in chapter 5. Let's take a look at this picture. We have for example, we have two homologous chromosomes. So let me see if I can find the pointer here. Okay, here's one homologous chromosome, and here's the other homologous chromosome. So remember, this is just two chromosomes because each chromosome itself consists of sister chromatids, right? So for example, here, this homologous chromosome, this comes from the father, it's blue. This one comes from the mother of this individual that's going to make gametes. And then what happens is you have the formation of a tetrad. The phenomenon is called synapsis. When you have synapsis of homologous chromosomes, the sister chromatids actually are able to exchange genetic material. When does this occur? Well, this occurs in prophase 1 of meiosis. Recall that meiosis has two stages, meiosis 1 and meiosis 2. Okay, and we can actually see this in this picture. Let me see if I can point at it. So, okay, we have two non-homologous chromosomes that are very close together, and they actually trade some genetic material. So this sister chromatid here, which is blue, picks up a bit at the very end that's red. This one that's red, because it came from the mother, picks up a bit that's blue. And so here now are the recombinant daughter chromosomes here, where they've split up. So potentially, if you imagine if there was a gene, for example, at this location on this chromosome, and there was a gene previously on the blue bit here, well, now it's separated. Now it's moved to a different gene. So now we've had essentially recombination, and this allows more variety in the gametes that can be formed. This slide illustrates the idea that when two genes are close together, recombination is rare. So we have genes A and B. As you can see here, A and B, they're close together. So for example, we have big A and big B here on this chromosome that comes from the father. That's why it's blue. Here, this chromosome that comes from the mother, it has a small a and a small b. And so let's imagine that there's a crossing over event, but it occurs at such a place, for example, here, such that the genes remain linked together. So in this case, only two types of gametes are made with respect to these genes, A and B. So recombination between two genes that are located close together is rare. Only two types of gametes are produced, even though this is a dihybrid cross. In this slide, we're going to explore what happens when two genes are further apart. Well, this is going to essentially make recombination more frequent. So here we have gene A and gene B, as you can see here. They're quite a bit further apart than the previous time. So now when we have a crossing over event, we can have a recombination. So crossing over doesn't happen all the time. In fact, it's relatively rare. So in general, this individual would make these gametes. It would have where the big B are still linked, little a, little b are still linked. But so 97% of the time that would happen, you'd produce these gametes, but there is some recombination that occurs. So for example, here, there's a 3% chance that you'll have big A and then little b, and little a and then big B. Right, so what happens here, because of recombination, instead of forming two gametes, we actually form 4 different gametes. So this allows for more variability when this gamete would then get together with another gamete, and then you'd have the zygote. So recombinant gametes are produced in a reduced proportion compared to non-recombinant. So that's shown here, 3% versus 97%. Now, it could be different. If the genes were really far apart, it could be as high as 5 or even 10% of recombinant gametes. So I'm going to end this talking about how this gene linkage and the idea of crossing over was actually quite helpful in the past. So in the past, the occurrence of crossing over was useful because it could tell us a little bit about the sequence of genes on a chromosome. So crossing over occurs more often between distant genes compared to genes that are closer together, as I illustrated earlier. And if you studied this with a lot of in a lot of detail and looked at many pairings, for example, you could actually get a good idea about exactly how far apart the genes were. Now, of course, nowadays we don't really have to do that anymore because it's possible to sequence the genes on these chromosomes, but it's still a useful idea at least. So if crossing over occurs frequently, then we know the genes are distant on the chromosome. If crossing over very rarely occurs, then we know that the genes are located close together on a chromosome. Okay, that's it for this video.